So we're continuing now on to the next session of the lecture. Right, so we're on adenovirus. That uses the second strategy. It initially is taken up by receptor-mediated endocytosis. As the endosome matures and becomes more acidic, the virus undergoes multiple uncoating steps in which structural proteins are sequentially moved from the capsid. Some of these steps require the action of a viral protease, which is inactive in the extracellular virus particle, probably because of interchanged disulfide bonds, but which is activated in the reducing environment of the endosome. One of the proteins released from the capsid lyses the endosomal membrane, releasing the remainder of the virus into the cytosol. This trimmed down virus then docks out of the nuclear pore complex, and the viral DNA genome is released through the pore into the nucleus, where it is transcribed. In these various entry strategies, viruses exploit a variety of host cell molecules and processes, including cell surface components, receptor-mediated endocytosis, and endosomal maturation steps. These strategies again illustrate the sophisticated ways that pathogens have evolved to utilize the basic cell biology of their hosts. Right? Now the next topic will be bacteria that enter host cells by phagocytosis. Bacteria are much larger than the viruses, and they are too large to be taken up by receptor-mediated endocytosis. Instead, they enter host cells through phagocytosis. Phagocytosis of bacteria is a normal function of macrophages. They control the tissues of the body and ingest and destroy unwanted microbes. Some pathogens, however, have acquired the ability to survive and replicate within macrophages after they have been phagocytosed. To the topic of tuberculosis is a serious lung infection that is widespread in some urban populations. Is caused by one such pathogen, Mycobacterium tuberculosis. This bacterium is usually acquired by inhalation into the lungs, where it is phagocytosed by alveolar macrophages. Although the microbe can survive and replicate within macrophages, the macrophages of most healthy individuals, with the help of the adaptive immune system, contain the infection within a lesion called a tubercle. In most cases, the lesion becomes walled off within a fibrous capsule that undergoes calcification, after which it can easily be seen on an X-ray of the lungs of an infected person. An unusual feature of tuberculosis is its ability to survive for decades within macrophages contained in such lesions. Later in life, especially if the immune system becomes weakened by disease or drugs, the infection may be reactivated, spreading in the lungs and even to other organs. Tuberculosis has been an obvious presence in human populations for thousands of years, but another bacterium that lives within alveolar macrophages was first recognized as a human pathogen only in 1976. Legionella pneumophila is normally a parasite of freshwater amoeba, which take it up by phagocytosis. When droplets of water-containing L. pneumophila or infected amoeba are inhaled into the lung, the bacteria can invade and live inside alveolar macrophages, which to the bacteria must seem just like large amoebae. This infection leads to a type of pneumonia known as Legionnaire's disease. The pathogen can efficiently spread by central air conditioning systems, as the amoebae that are the bacterium's normal host are particularly adept at growing in air conditioning cooling towers. Moreover, these cooling systems produce microdroplets of water that are easily inhaled. The incidence of Legionnaire's disease has increased dramatically in recent decades, and outbreaks are frequently traced to the air conditioning systems in office buildings, hospitals, and hotels. Other forms of modern aerosolization, including decorative fountains and produce sprayers and supermarkets, have also been implicated in outbreaks of this disease. Uptake of Legionella pneumophila by a human mononuclear macrophage. This electron micrograph shows the unusual coil structure induced in the surface of the phagocyte by the bacterium. Some bacteria invade cells that are normally non-phagocytic. One way that bacteria can induce such a cell to phagocytose them is by expressing an adhesion that binds with a high affinity to a cell adhesion, a protein that the cell normally uses to adhere to another cell or to the extracellular matrix. For example, a bacterium that causes diarrhea, Yersinia pseudotuberculosis, a close relative of the plague bacterium Yersinia pestis, expresses a protein called invasion that binds to beta-1 integrins and Listeria monocytogenes, which causes a rare but serious form of blood poisoning, expresses a protein that binds to E-cadherin. Binding to these transmembrane adhesion proteins fools the host cell into attempting to form a cell junction. And it begins moving actin and other cytoskeletal components to the site of bacterial attachment. Since the bacterium is small relative to the host cell's attempt to spread over the adhesive surface of the bacterium results in the phagocytic uptake of the bacterium, a process known as zipper mechanism of invasion. You can see that in the figure here. The zipper mechanism. Okay, so it moves across. Okay, that's interesting. And the similarity of this form of invasion to the natural process of cell adhesion was underscored by the determination of the three-dimensional structure of invasion. This base bacterial protein has an RGD motif, the structure of which is almost identical to that of the normal beta-1 integrin binding site in laminin. Mechanisms used by bacteria to induce phagocytosis by non-phagocytic host cells. The zipper and B trigger mechanisms for pathogen-induced phagocytosis both require polymerization of actin at the site of bacterial entry, frames from a time lapse. More on that in this diagram coming up, right? Frames from a time lapse movie recorded 10 seconds apart, showing the formation of a giant membrane ruffle arrow. It's there in the arrow, right? When Salmonella enterica contacts a host cell in culture, this leads to the phagocytosis of the bacterium by the trigger mechanism. That's interesting to note. Okay, going back to the subject proper. Right. A second pathway by which bacteria can invade non-phagocytic cells is known as the trigger mechanism. And it is used by various pathogens including Salmonella enterica, which causes food poisoning. This dramatic form of invasion is initiated when the bacterium injects a set of effector molecules into the host cell cytoplasm through a type 3 secretion system. Some of these effector molecules activate Rho family GTPases, and which stimulates actin polymerization. Others interact with cytocellular elements more directly and severing actin filaments and causing the rearrangement of cross-linking proteins. The net effect is to cause dramatic localized ruffling on the surface of the host cell, which throws up large actin-rich protrusions that fold over and trap the bacterium within large endocytic vesicles called macropinosomes. The overall appearance of cells being invaded by a trigger mechanism is similar to the dramatic ruffling induced by some growth factors, suggesting that similar intracellular signaling cascades are activated in both cases. Intracellular parasites actively invade host cells. The uptake of viruses by receptor-mediated endocytosis and bacteria by phagocytosis requires energy probed by the host cell. The pathogen is a relatively passive participant, usually providing a trigger to initiate the invasion process but not contributing any metabolic energy. In contrast, the invasion of intracellular eukaryotic parasites, which are typically much larger than the bacteria, proceeds through a variety of complex pathways that usually require significant energy expenditure on the part of the parasite. Toxoplasma gondii, the cat parasite that causes occasional serious human infection, is an instructive example. When this protozoan contacts a host cell, it protrudes a microtubule rib structure called a conoid and then reorients so that the conoid is touching the host cell surface. The parasite then slowly pushes its way into the host cell. The energy for invasion seems to come entirely from the parasite, and the process can be disrupted by depolymerizing the parasites, but not the host actin cytoskeleton. As the parasite moves into the host cell, it secretes lipids from a pair of specialized organelles, generating a vacuolar membrane that is made primarily of parasite lipids and proteins. In this way, the organism protects itself in a membrane-enclosed compartment within the host cell that does not participate in host cell membrane trafficking pathways and does not fuse with lysosomes. The specialized vacuolar membrane allows the parasite to take up metabolic intermediates and nutrients from the host cell cytosol, but excludes larger molecules. The life cycle of the intracellular parasite Toxoplasma gondii. After attachment to a host cell, T. gondii secretes lipids, red, from internal organelles. After invasion, the parasite replicates the special vacuole formed from parasite lipids. An entirely different but no less peculiar invasion strategy is used by the protozoan Trypanosoma cruzi, which causes a multi-organ disease, Chagas disease, mainly in Mexico and Central and South America. After reattachment to beta-1 integrins on the host cell surface, this parasite induces a local elevation of calcium in the host cell cytoplasm. The calcium signal recruits lysosomes to the sites of parasite attachment, which then fuse with the plasma membrane, allowing the parasite to enter a vacuole derived most entirely from lysosomal membrane. As we discussed below, most intracellular pathogens go to great lengths to avoid exposure to the hostile proteolytic environment of the lysosome, but this trypanosome uses this lysosome as its means of entry. In the vacuole, the parasite secretes a transilidase enzyme that removes salic acid from lysosomal glycoproteins and transfers it to its own surface molecules, thereby coating itself with host cell sugars. Next, the parasite secretes a pore-forming toxin that lyses the vacuolar membrane, releasing the parasite into host cell cytosol, where it replicates. Invasion of the Trypanosome cruzi, this parasite recruits host cell lysozymes to its site of attachment. The lysosome fuse with the plasma membrane to create a vacuole. The lysosomes fuse with the plasma membrane to create a vacuole constructed almost entirely of lysosomal membrane. After a brief stay in the vacuole, the parasite secretes a protein porin that disrupts the vacuolar membrane, allowing the parasite to escape into the host cell cytosol and replicate. Many pathogens alter membrane traffic in the host cell. The two examples of intracellular parasites discussed in the preceding section raise a general problem that faces all pathogens with an intracellular lifestyle, including viruses, bacteria, and parasites. They must deal in some way with the membrane traffic in the host cell. After endocytosis by a host cell, they usually find themselves in an endosomal compartment that normally would fuse with the lysosomes. They therefore must either modify the compartment to prevent its fusion, escape from the compartment before getting digested, or find ways to survive in the hostile environment of the phagolysosome. The choice is faced by an intracellular pathogen after entry, generally through endocytosis or phagocytosis, into a membrane-enclosed compartment. Intracellular pathogens may use one of three strategies to survive, replicate. Pathogens and the follow strategy of 1. Include all viruses, Trypanosoma cruzi, Listeria monocytogenes, Shigella flexneri. Those that follow strategy include Plasmodium falciparum, Mycobacterium tuberculosis, Salmonella enterica, Legionella pneumophilia, and Chlamydia trachomatis. Those that follow strategy 3 include Coxiella burnetii and Leishmania. Now, most pathogens use either the first or second strategy, it is much less common to find pathogens that can survive in the phagolysis zone, as we have seen. Trypanosoma cruzi, uses the escape route, as do essentially all viruses. The bacterium Listeria monocytogenes also uses this strategy. It is taken up into cells via the zipper mechanism discussed earlier and secretes a protein called hemolysin that forms large pores in the phagosomal membranes and eventually disrupting the membrane and releasing the bacteria into the cytosol. Once in the cytosol, the bacteria continue to secrete hemolysin, but it does not destroy the plasma membrane. The hemolysin selectively destroys only the phagosomal membrane. The bacterium Listeria monocytogenes also uses this strategy. It is taken up into cells via the zipper mechanism and secretes a protein called hemolysin that forms large pores in the phagosomal membrane, eventually disrupting the membrane and releasing the bacteria into the cytosol. Once in the cytosol, the bacteria continue to secrete hemolysin, but it does not destroy the plasma membrane. The hemolysin selectively destroys only the phagosomal membrane for two reasons. First, it is 10 times far more active at the acidic pH found in the phagosome than at neutral pH found in the cytosol. And second one, hemolysin contains a PES sequence, a peptide motif rich in proline, glutamine, serine, and threonine, which is recognized by the protein degradation machinery of the host cell, leading to the rapid degradation of the bacterial hemolysin, in the proteasome. The hemolysin is stable in the phagosome as the degradation machinery does not have access to there. The hemolysin secretion by L monocytogenes is closely related to hemolysin secretion by other bacteria and that are not intracellular pathogens. Figure 686 is this. Take a screenshot. Figure 2529 is this. Take a screenshot. Moving on. These related hemolysins, however, all lack PES sequence. It seems that the L-monocytogenes hemolysin is acquired in essentially eukaryotic protein domain expressly to allow its activity to be regulated in the host cell. The selective destruction of the phagosomal membrane by Listeria monocytogenes. L-monocytogenes attaches to E cadherin on the surface of epithelial cells and induces its own uptake by the zipper mechanism. By far the most common strategy used by intracellular bacteria and parasites is to modify the endosomal compartment so that they can remain there and replicate. They must modify the compartment in at least two ways. First, they must prevent lysosomal fusion, and second, they must provide a pathway for importing nutrients from the host cytosol. Different pathogens have distinct strategies for doing this. As we have seen, Toxoplasma gondii creates its own membrane enclosed compartment that does not participate in normal host cell membrane traffic and allows nutrient transport. This option is not open to bacteria, which are too small to carry in their own supply membrane. Mycobacterium tuberculosis somehow prevents the very early endosome that contains it from maturing so that it never acidifies or acquires the characteristics of a late endosome. Endosomes containing Salmonella enterica, in contrast, do acidify and acquire markers of late endosomes. But they arrest their maturation at a stage prior to lysosomal fusion, and other bacteria seem to find shelter in intracellular compartments that are completely distinct from the usual endocytic pathway. Legionella pneumophili, for example, replicates in compartments that are wrapped in layers of rough endoplasmic reticulum. Chlamydia trachomatis is a sexually transmitted bacterial pathogen that can cause sterility and blindness, replicates in a compartment that seems to be most similar to part of the exocytic pathway. The mechanisms used by these organisms to alter their membrane compartments are not yet understood. Modifications of intracellular membrane trafficking by bacterial pathogens. For intracellular bacterial pathogens, Mycobacterium tuberculosis, Salmonella enterica, Legionella pneumophilia, and Chlamydia trachomatis all replicate in membranes enclosed. Take a screenshot of this, and the continuation all replicate in membrane enclosed compartments, but in four compartments differ from M. tuberculosis remains in a compartment that has early endosomal markers and continues to communicate with the plasma. Membrane S. enterica replicates in a compartment that has endosomal markers. It has late endosomal markers and does not communicate with the surface. L. pneumophilia replicates in unusual compartment that is wrapped in several layers of rough endoplasmic reticulum membrane. C. trachomatis replicates in an exocytic compartment that fuses with the vesicles coming from the Golgi apparatus. And returning to subject matter proper. Viruses also alter membrane traffic. The host cell enveloped viruses must acquire the membrane from host cell phospholipids. In the simplest cases, virally encoded proteins are inserted into the ER membrane and follows the usual path through the Golgi apparatus to the plasma membrane, undergoing various post-translational modifications en route. The viral capsid then assembles the plasma membrane and buds out from the cell surface. This is the mechanism used by HIV. Other enveloped viruses interact in complex ways with membrane trafficking pathways in the host. Even some non-enveloped viruses alter membrane traffic in the host cell to suit their own purposes. The replication of poliovirus, for example, is carried out by a membrane-associated virus encoded RNA polymerase. Replication can proceed more quickly if the surface area of the host cell membrane is increased. To accomplish this, the virus induces increased lipid synthesis in the host cell and blocks secretion from the ER. Intracellular membranes thereby accumulate, expanding the surface area on which the RNA replication can occur. Complicated strategy for viral envelope acquisition. Herpes virus capsids are assembled in nucleus and then bud through the inner membrane, acquiring a member coat. They then apparently lose coat, which they confuse with the outer membrane. Intracellular membrane alterations induced by a poliovirus protein. Poliovirus, like other positive stranded RNA virus, replicates its RNA genome using a polymerase that associates with intracellular membranes. And to detail that's the figure for your screenshots and the written details. They then apparently lose their coat when they fuse with the outer mycelial membrane to escape into the cytosol. Subsequently, they bud into Golgi apparatus and bud out again on the other side, acquiring two new membrane coats. The virus then buds from the cell with a single membrane when its outer membrane fuses with the plasma membrane. Vacciinovirus, which is closely related to the virus that causes smallpox, is assembled in replication factories deep with the cytosol. The first structure assembled contains two membranes acquired from the Golgi apparatus by a poorly defined wrapping mechanism. A variable proportion of these viral particles are then engulfed by the membranes of a second intracellular membrane enclosed compartment. These viral particles have a total of 4 layers of membrane envelope. After fusion of the plasma membrane, the virus escapes with these 3 layers. And the next one. The poliovirus, like other positive stranded RNA viruses, replicates its RNA genome using a polymerase that associates with intracellular membranes. Several other membranes encoded in its genome alter the structure and dynamic behavior of the membrane-enclosed organelles in the host cell. These electron micrographs show a normal CORAH-7 cell left and a cell expressing the 3A protein from poliovirus right. In the transfected cell, the ER is swollen and traffic from the ER to the Golgi is inhibited. So that is for the screenshot there. Back to the subject. Now, the cytoplasm. Viruses and bacteria exploit the host cell's cytoskeleton for intercellular movement. The cytoplasm of mammalian cells is extremely viscous. It is crowded with organelles and supported by networks of cytoskeleton filaments, all of which inhibit the diffusion of particles the size of a bacterium or a viral capsid. If a pathogen must reach a particular part of a cell to carry out part of its replication cycle, it must move actively, as with transport of intracellular organelles. Pathogens generally use the cytoskeleton for active movement. Several bacteria that replicate in the host cell cytosol rather than in membrane-enclosing compartments have adopted a remarkable mechanism for moving, which depends on actin polymerization. These bacteria include Listeria monocytogenes, Shigella flexneri, and rickettsii, which causes Rocky Mountain spotted fever and induces the nucleation and assembly of host cell actin filaments at one of the pole of the bacterium. The growing filaments generate substantial force and push the bacteria through the cytoplasm at rates up to 1 micrometer per second. New filaments form at the rear of each bacterium and are left behind like a rocket trail as the bacterium advances, while depolymerizing again with a minute or so as they encounter depolymerization factors in the cytosol. When a moving bacterium reaches the plasma membrane, it continues forward to move outward, inducing the formation of a plug. Right, and inducing the formation of a long thin protrusion with the bacterium as this projection is often engulfed by a neighboring cell, allowing the bacterium to enter the neighbor cytoplasm with exposure to the extracellular environment, thereby avoiding recognition by antibodies produced by the host adaptive immune mechanism. The actin-based movement of Listeria monocytogenes within and between host cells. These bacteria induce the assembly of actin-rich tails in the host cell cytoplasm, which enable them to move rapidly. Motile bacteria spread from cell to cell by forming note these diagrams properly, right? These bacteria induce the assembly of actin-rich tails in the cytoplasm. Right. And which enable them to move rapidly. Motile bacteria spread from cell to cell by forming membrane-enclosed protrusions that engulf the neighboring cells. Fluorescence micrograph of the bacteria moving in a cell that has been stained to reveal both bacteria and actin filaments. Note the comet-like tail of actin filaments green, behind each moving bacteria red. Regions of overlapping red and green fluorescence appear yellow. That's interesting. You see that? The yellow is the bacteria, the green are filaments. Interesting. Right, moving back to the subject. The molecular mechanism of pathogen-induced actin assembly has been determined for these two of these bacteria. The mechanisms are different, suggesting that they evolved independently, although both make use of the same host cell regulatory pathway that normally controls the nucleation of actin filaments. They exploit different points in the pathway, as discussed, and then in the chapter-wise discussion ensues. For activation of the small GTPase CDC42 by growth factors or other external signals causes the activation of a protein called NWASP, which in turn activates the ARP complex that can nucleate the growth of the new actin filament. An L-monocytogen surface protein directly binds to and activates the ARP complex to initiate the formation of an actin tail, whereas an unrelated surface protein S. flexneri binds to and activates NWASP, which then activates the ARP complex. Remarkably, vaccinia virus uses yet another mechanism to move intracellularly by inducing actin polymerization, again exploiting the same regulatory pathway. There are molecular mechanisms for actin nucleation by various pathogens. The bacterial Listeria monocytogens and Shigella flexneri and the virus vaccinia all move intracellularly using actin polymerization. To induce actin nucleation, all of these pathogens recruit more take a screenshot. To induce actin nucleation, all of these pathogens recruit and activate the ARP complex, although each pathogen uses a different recruitment strategy. And the ARP complex, you can just check it on the figure. Right? Check this, take a screenshot, okay? And we're back to the subject. L-monocytogens expresses a surface protein 8A that directly binds to and activates the ARP complex. X-flexneri expresses surface protein ICSA and related to ACTA that recruits a host cell signaling protein, NWASP. NWASP in turn recruits the ARP complex along with the other host proteins, including WIP, WASP, interacting protein. Vaccinia virus expresses an envelope protein that is phosphorylated on tyrosine by a host cell protein tyrosine kinase. The phosphorylated protein then recruits NCK, which binds WIP. WIP binds NWASP, which recruits and activates the ARP complex. The more complicated cascade used by vaccinia is thought to closely resemble how the ARP complex is activated by biochemoactive factors in motile eukaryotic cells. Despite these distinct molecular strategies, the actin comet tail is formed by all three pathogens that we're familiar. The pathogens move at similar speeds inside affected cells. Back to the subject matter proper. The pathogens rely primarily on microtubule-based transports and move within the host cell. This movement is particularly well illustrated by viruses that infect neurons. An important example is provided by the neurotropic alpha herpes virus, a group that includes the virus that causes chickenpox. Upon infection of sensory neurons, the virus particles are transported to the nucleus by microtubule-based transport, probably mediated by attachment of the capsids to the motor protein dynein. After replication assembly in the nucleus, the enveloped virus is transported along microtubules away from the neural cell body down the axon, presumably attached to a kinesia protein, as you can see on this figure. Right? Okay. Take a screenshot. This nerve cell has been infected with alpha herpes virus that has been genetically engineered. I think you can see the figures, right? To express green fluorescent protein GFP used to one of the capsid proteins. In this segment of action, several viral particles are visible, and two of them numbered 1 and 3 are moving away from the cell body. Right? One bacterium that is known to associate with microtubules is Wolbachia. This fascinating genus includes many species that as parasites or symbionts of insects and other invertebrates living in the cytosol, which the animal infection is spread vertically from mother to offspring. As it was, Wolbachia are also present in eggs. The bacteria ensure their transmission into every cell by binding to tubules, and they are therefore segregated by the mitotic spindle simultaneously with chromosome segregation when infected or cell divides. Fluorescence micrograph. Wolbachia red associated with microtubules green and of mitotic panel in Drosophila embryo. The clumps of bacteria at the spindle poles will be segregated into two dotted cells when each infected cell divides. Check this, take a screenshot. The clumps of bacteria at the spindle pole will be segregated into two dotted cells when each infected cell divides. Viruses take over the metabolism of the host cell. Most intracellular bacteria and parasites carry the basic genetic information required for their own metabolism and replication, and they rely on their host cells for oligonutrients. Viruses, in contrast, use the basic host cell machinery for most aspects of the reproduction. They all depend on host cell ribosomes to produce their proteins, and some also use host cell DNA and RNA polymerases for replication and transcription, respectively. Many viruses encode proteins that modify the host transcription or translation apparatus to favor the synthesis of viral proteins over those of the host cell. As a result, the synthetic capability of the host cell is directed principally to the production of new virus particles. For example, encodes a protease that specifically cleaves the HEA binding factor component or TFID, effectively shutting off all host cell transcription via RNA polymerase. 2. Influenza produces a protein that blocks both the splicing and the polyadenylation of mRNA transcripts, which therefore fail to be exported from the nucleus. Translation initiation of most host cell mRNAs depends on recognition of their 5' cap by a group of translation initiation factors. Translation initiation of host mRNAs is often inhibited during a viral infection so that the host cell ribosomes can be used effectively for synthesis of viral proteins. Some viruses encode endonucleases that cleave the 5' cap from the host cell mRNAs. Some even then use the liberated 5' caps as primers to synthesize viral mRNAs, a process called cap snatching. Several other viral RNA genomes encode proteases that cleave certain translation initiation factors. These viruses rely on 5' cap independent translation of the viral RNA using internal ribosome entry sites, IRESs. A few DNA viruses use host cell DNA polymerase to replicate the genome. These viruses need to solve a problem. DNA polymerase is expressed at high levels only during the S phase of the cell cycle, whereas most cells that the virus infects spend most of their time in G1 phase. Adenovirus solves this problem by inducing the host cell to enter S phase. Its genome encodes proteins that inactivate both RB and P53, two key suppressors of cell cycle progression. As might be expected for any mechanism that induces unregulated DNA replication, these viruses are frequently oncogenic. Pathogens can alter the behavior of the host organisms to facilitate the spread of the pathogen. As already seen, pathogens can alter the behavior of the host cell in ways that benefit the survival and replication of the pathogen. Similarly, pathogens often alter the behavior of the whole host organism to facilitate the pathogen spread, as we saw earlier or Trypanosoma brucei and Yersinia pestis. In some cases, it is difficult to tell whether a particular host response is more for the benefit of the host or for the pathogen. Pathogens like Salmonella enterica that cause diarrhea, for example, usually produce self-limiting infections because the diarrhea efficiently washes out the pathogen. The bacterial-laden diarrhea, however, can spread the infection to a new host, Similarly, coughing and sneezing help to clear pathogens from the respiratory tract, but they also spread the infection to new individuals. A person with a common cold may produce 20,000 droplets in a single sneeze. All carry rhinovirus or coronavirus. Okay, that's an important note. A person with a common cold may produce 20,000 droplets in a single sneeze, all carrying rhinovirus or coronavirus. A frightening example of a pathogen modifying most behavior is seen in rabies. As first described in Egyptian writings over 3,000 years ago, this virus replicates in neurons and causes infected people or animals to become rabid. They are unusually aggressive and develop a strong desire to bite. The virus is shed in the saliva and transmitted through the bite wound into the bloodstream of the victim, spreading the infection to a new host. Toxoplasma gondii, a eukaryotic parasite that forms lesions in the muscle and brain tissue, provides an equally remarkable example. It can complete the life cycle only in its normal host, cats. If it infects an intermediate host, such as a rodent or a human, the infection is a dead end for the parasite, unless the intermediate host is eaten by a cat. Behavioral studies show that rats infected with G. gondii lose their innate fear of cats and instead preferentially seek out locations perfumed with the cat urine over locations perfumed with rabbit urine, exactly opposite of normal rat behavior. But the most dramatic example of pathogens modifying host behavior belongs to Wolbachia. These bacteria manipulate the sexual behavior of the host to maximize their dissemination, as described earlier. Wolbachia are passed vertically into offspring through eggs. If they live in a male, however, they hit a dead end, as they excluded from sperm. And some species of Drosophila Wolbachia bountify the sperm of their host so that they can fertilize the eggs only of infected females. This modification creates a reproductive advantage for infected females over the uninfected females, so that the overall proportion of Wolbachia carriers increases. In other host species, a Wolbachia infection kills males but spares females, increasing the number of females in the population and thus the number of individuals that can produce eggs to pass on to infection. De-infection. In a few types of wasp Wolbachia infections enable the females to produce eggs that develop pathogenetically without the need for fertilization by sperm. And the species, males have been completely eliminated. For some its hosts, Wolbachia has become an indispensable symbiont, and curing the infection causes death of the host. In one case, humans are making use of this dependence. The filarial nematode that causes African liver blindness is difficult to kill with antiparasitic medications, but when people with liver blindness are treated with antibiotics that cure the nematode Wolbachia infection, the nematode infection is also arrested. That's interesting. Pathogens evolve rapidly. The complexity and specificity of the molecule interactions between pathogens and their host might suggest that virulence would be difficult to acquire by random mutation. But new pathogens are constantly emerging and old pathogens are constantly changing in ways that make familiar infections difficult to treat. Pathogens have two great advantages that enable them to evolve rapidly. First, they replicate very quickly, providing a great deal of material for the engine of natural selection. Whereas humans and chimpanzees have acquired a 2% difference in a genome sequence of about 8 million years of divergent evolution. Poliovirus manages a 2% change in its genome in 5 days, about the time it takes the virus to pass from the human mouth to the gut. Second, this rapid genetic variation is acted on by powerful selective pressures provided by the host adaptive immune system and by modern medicine, which destroy pathogens that fail to change. A small-scale example of the constant battle between infection and immunity is phenomenon of antigenic variation, which is described as the ability to change the antigens displayed on the cell surface, a property of some pathogenic microorganisms. that enables them to evade attacks by the immune system. All right, and an important adaptive immune response against many pathogens is the production of antibodies that recognize specific antigens on the pathogen surface. Many pathogens evade complete elimination by antibodies by changing these antigens during the course of an infection. Some parasites, for example, undergo programmed rearrangements of the genes encoding their surface antigens. The most striking example is in African trypanosomes such as Trypanosoma brucei, a parasite that causes sleeping sickness and is spread by an insect vector. T. brucei is a close relative of T. cruzi, but it replicates extracellularly rather than inside cells. T. brucei is covered with a single type of glycoprotein called variant-specific glycoprotein, VSG, which elicits a protective antibody response that rapidly clears most of the parasites. The trypanosome genome, however, contains about 1,000 VS genes, each encoding a VSG with distinct antigen properties. Only one of these genes is expressed at any one time by being copied into an active expression site in the genome. The VSG gene expressed can be changed repeatedly by gene arrangements that copy new alleles into the expression site. In this way, a few trypanosomes with an altered VSG escape the antibody-mediated clearance, replicate, and cause the recurrence of disease, leading to a chronically cyclic infection. Antigenic variation in trypanosomes. There are about 1,000 VSG genes in Trypanosoma brucei, but only one site for VSG gene expression. An inactive gene is copied into the expression site by gene conversion, where it is converted by RAS switching. Events that allow the trypanosome to change repeatedly. The surface antigen it expresses. A person infected with trypanosomes expressing VSG quickly mounts protective antibody response, which results in clearance of most of the parasites expressing this antigen. However, a few of the trypanosomes may have switched to expression of VSG, which can now proliferate until they are cleared by the anti-VSG antibodies. By the time, however, some parasites will have switched to VSG, and so the cycle repeats seemingly indefinitely. A different type of antigenic variation occurs during the course of infection by viruses that have error-prone replication mechanisms. Retroviruses, for example, acquire an average 1-point mutation every replication cycle because the viral reverse transcriptase that produces DNA from the viral RNA genome cannot correct nucleotide misincorporation errors. A typical untreated HIV infection may eventually involve HIV genomes with every possible point, right? Retroviruses, for example, acquire an average 1-point mutation every replication cycle because the viral reverse transcriptase that produces DNA from the viral RNA genome cannot correct nucleotide and misincorporation errors. A typical untreated HIV infection may eventually involve HIV genomes with every possible point mutation. In some ways, the high mutation rate is beneficial for the pathogen by a microevolutionary process of mutation and selection within each host. The virus can switch from infecting macrophages to infecting T cells, and once treatment is begun, it can quickly acquire resistance to drugs. If the reverse transcriptase error rates were too high, however, deleterious mutations might accumulate too rapidly to follow the virus to survive. Furthermore, rapid viral diversification in one host does not necessarily lead to rapid evolution of the virus in the population, as a mutated virus may not be able to infect a new host. For HIV-1, the extent of this constraint can be estimated by examining the sequence diversity among different individuals infected with the virus. Remarkably, about 1/3 of the nucleotide position coding sequence of virus and invariant nucleotide sequence, some parts of the genome, such as the N gene, can differ by 30%. This extraordinary genomic plasticity greatly complicates attempts to develop vaccines against HIV and lead to rapid drug resistance. Further consequence has been rapid diversification and emergence of a new HIV strain. Sequence comparison between various strains of HIV, very many similar immunodeficiency viruses, IV, simian immunodeficiency virus from a variety of different monkey species, seem to indicate virulent type of HIV. HIV-1 may have jumped from chimpanzees to humans as recent. That's the diagram there. Diversification of HIV-1, HIV-2, and related strains of HIV genetic distance. With any two viral isolates is found by following their shortest path. Joining them in the tree, HIV-1 is divided into two groups, major M and outlier O, HIV-1M. Group is responsible for the global AIDS epidemic. HIV-M is further subdivided in subtypes. Or through A through G, subtype B is dominant in America and Europe, B, C, and E predominant in Asia. All subtypes are found in Africa. At least two monkey viruses, chimpanzee and mandrill, are more closely related to HIV-1 than is HIV-2, suggests that HIV-1 and HIV-2 arose independent of one another. HIV-1 SLV-CPZ chimpanzee are estimated to have diverged about 1930. This tree was constructed from the nucleotide sequence of the GAG gene, using a database containing 16,000 sequences from virus samples isolated around the world. And the subject matter proper. Rapid evolution of bacteria frequently takes place by horizontal gene transfer rather than by point mutation. Most of the horizontal gene transfer is mediated by the acquisition of plasmids and bacteriophages. Bacteria readily pick up pathogenicity islands and virulent plasmids. Once a bacterium has acquired a new set of virulence-related genes, it may quickly establish itself as a new cause of human epidemics. Yersinia pestis, for example, is an infection endemic to rats and other rodents. It first appeared in human history in 542 when the city of Constantinople was devastated by plague. Sequence comparison of Y. pestis to its close relative Y. pseudotuberculosis, which causes a severe diarrheal disease, suggests that Y. pestis may have emerged as a distinct strain only a few thousand years ago, not long before its debut in the field of urban devastation. The emergence and evolution of new infectious disease is in many cases exacerbated by changes in human behavior. For example, crowded and filthy living conditions in medieval cities contributed to the spread of plague to humans from the natural rodent host. The tendency of modern humans to live at high population densities in large cities has also created the opportunity for infectious organs to initiate epidemics such as influenza, tuberculosis, and AIDS, which could not have spread so rapidly or so far among sparser human populations. Air travel can, in principle, allow an asymptomatic, newly infected host to carry epidemic to any previously unexposed population within a few hours or days. Modern agricultural practice also fostered the emergence of certain types of infectious agents that could not have easily developed in the hunter-gatherer societies of early humans. Influenza viruses, for example, are unusual in that their genome consists of several, usually 8 strands of RNA. When two strains of influenza infect the same host, the strands of the two strains can reshuffle to form a new and distinctive type of influenza virus. Prior to 1900, the influenza strain that infected humans caused a very mild disease. A separate influenza strain infected fowls, ducks, chickens, but could not infect humans. Avian human strains are capable of infecting pigs, can recombine pig hosts to form strains, cause serious human disease. Communities with pigs are farmed together with chickens or turkeys. Newly recombined influenza strains periodically emerge and cause worldwide epidemics. The first and most serious epidemic was the Spanish flu in 1918, which killed people more than World War I. Drug-resistant pathogens are growing problems. While some human activities have promoted spread of certain infectious disease, advances in public sanitation, medicines have prevented or alleviated the suffering caused by many others. Effective vaccines and worldwide vaccine programs have eliminated smallpox and severely reduced poliomyelitis. Many deadly child infections, as mumps, measles, and are now rarities in wealthy industrialist nations. However, there are still many widespread and devastating infectious diseases such as malaria, for which no vaccines are available. Of equal importance is the development of drugs that cure rather than prevent infections. The most successful class are the antibiotics, which kill bacteria. Penicillin was one of the first antibiotics used to treat infections in humans, introduced into clinical use just in time to prevent the tens of thousands of deaths from infectious battlefield wounds in World War II. Rapid evolution of pathogens is enabled to target bacteria to develop resistance to antibiotics. Typical lag between introduction of antibiotic into clinical use and the appearance of resistant strains is only a few years. Similar drug resistance arises among viruses when infections are treated with antiviral agents. Virus populations in HIV-infected persons treated with the reverse transcriptase ACT are examples that require complete resistance to the drug within a few months. The current protocol for treatment of HIV involves simultaneous use of 3 drugs, which helps minimize acquisition of resistance.